Yes, now I invite our colleague Dr. Abhishek Rathore to share his ideas and thoughts about the data curation as well as the data management, breeding data management. Thank you.
So, my name is Abhishek Rathore. I work as a biometrician with ICRISAT, and my main job is to help scientists across all disciplines, across all locations in planning of their studies, and those studies should have statistical soundness. So so that may belong to any of the area. Like that may be a socio-economic study, that may be a breeding study, that may be a soil science study, on-farm trials. Statistics plays a major role in most of the places. So we try to help researchers how to plan the study in a way that it should be statistical sound. Second, when the study has been designed, data has been collected, sometimes protocol of data collection also comes into the domain. And when data is collected, how do the proper analysis, and then after analysis it comes to data archival. So this is a whole story where we are involved, and we take help of data management unit and other colleagues. By the way, you need some break for a few minutes or it's okay? Okay, yeah, because I think from one, so like I'll go slow and no presentation. I like the mode in which Chukka was communicating with everybody. You can ask any question anytime. We won't wait till last because I may end abruptly. So anytime you can throw your questions to me and I'll be happy, because this is I have seen the participants list and I hope this is a gathering in which many diverse field persons. It's a very, very good, very nice diverse group. But with my limited knowledge in statistics and in breeding, I'll try to focus mainly on the crop things. But I'll be happy if you can ask question to your domain and I might try to. But before continue, I would like to say one thing. A question was asked to Chukka regarding incentives to scientists for submitting data. So I think two things are very clear. Well, this is my personal views, and they may be in the official documentation also soon with ICRISAT. First thing, that we all are being paid for the job. None of us is doing charity. Well, we all have a mission, because it is not only to earn money we are working, because we might have better jobs to do, but we all have a mission, as our Director General always says, to elevate the poverty level and status of the farmers for whom we are working. So that is one way. But the another, whatever donors, I think the biggest incentive what we get when we see that our research is being taken in practice, people are using that research, our variety is being released. I think there is no big incentive to a scientist better than that. And no money, no monetary benefit and other things can really match to those things when we see that kind of things. But I also understand that concern what was raised regarding the, I shouldn't say stealing, like using of your data by somebody else. So if we really have not analyzed our data in 5 years, that means we are not really going to analyze that. So if somebody else is analyzing that data before us after 5 years, is a good guy. At least he is helping the money what donor has put into the. So I think it's a good way if somebody is analyzing our data. The sharing has power. Since we shared, the Chukka has shown the dataverse. So dataverse, like, you need not, all data will not be open. I don't know, I was late, so I don't know. Chukka has shown that few data sets are restricted. You click, you will only get the information. So just I wanted to compliment, like, I really enjoyed the discussion, because we had these kind of discussions earlier with the Gates Foundation. They also wanted to incentivize the process by monetary benefit by some other things, but all scientists say that really don't require money from ICRISAT. It's the credit what goes. So we have taken that into account. Whatever data Chukka is going to share, it will come, the name of the scientist will always go through. And the who generated, the contributor, donor. So metadata we have selected very nicely, very carefully in that regard. Okay, so now I'll start presentation. Okay, how many of you have agricultural background? Oh, good number. How many of you are from breeder, soil scientist, agronomist, anywhere related to crop or maybe the animals? Okay, quite a good number. I hope around 40% people. And how many of you have ever published your research work? Ever published your research work? How many of you? Only 20%? I think it should be more. Okay. And how many of you ever use statistics for your publication? Yes, it's okay. No, just I wanted to check, madam. So, so everybody has used statistics. Okay. Okay. No. But at least, okay. So because today what I'm going to discuss is research data curation, validation, analysis. The title is very big, analysis will be short. Because I cannot cover those three things in 1 hour, maybe in 1 week we can do. So the data curation I'll talk mainly, that what is data curation, how we do it and how it affects. I'll take an example from a breeding perspective, but same, my discussions will be from any field. So I'll talk for a data which has 150 progenies and it was a RCBD design. How many could not understood this? Okay, from informatics specialist I understand it is difficult. So I'll explain more. We have around 150 different identities. They may be the human, they may be the animals, they may be the plants, they may be the object of our in which we are interested. Maybe the metallic plate, maybe the silicon wafer, maybe the some electronic component, maybe the brands of tube lights. So just we have taken example, maybe the villages from where we have obtained the data, something like this. So I had a data set in which there were 150, those observations were there. And the experimental design, so the idea was to select which of them 150 is best. Most of the time we have this kind of, or which of them is worst. The village, which village out of our whole district what we have selected is performing well in terms of technological adoption. Then it will become a CRD well. And otherwise, so these may be our. So the usual process at that time comes when you want to compare several things. A F-test comes into the picture. We won't go into detail of that, okay? But the idea here was that we have 150 different plants which were showing different level of phenotypic variation. Like some of them were having very small fruit size, some of them were having very big fruit size, like we see in the market. Now idea was to detect the gene which is responsible for this up and down regulation. Is it clear or not? Most of us have read biology, isn't it? In biology we know there are genes. So these kind of studies were laid out to locate the position of gene which is influencing the phenotypic variation. Like few of us are very tall, few of us are shorter. So then there is a gene which regulates height. So we can take a sample of 150 people who show lots of gradient in the height or weight and anything, and then we can try to see their DNA and look for with a scanner, where is the gene. It's not as simple, but like you can do a regression and you can check that which gene is regulating. Okay. So to do that, first step is always analysis of variance, which we call ANOVA. Anybody who has never heard name of ANOVA? Everybody heard name of ANOVA? Don't be shy, I'll explain. ANOVA is a method in which we see the variation present in the data and try to see whether that variation is significant or not. Anyways, so I did the ANOVA. And this is a usual table for ANOVA, in which we have few source of variation, the first column. So since it was a RCBD design, then we had one block as source of variation, the RIL as source of variation, there is an error and as a total. So anybody who has some idea of statistics will be very happy to say that block effect was significant and real effect was significant. How they are significant? At 95%. Well, this value is less than 0.05, so we say it is significant, isn't it? Most of us know that. Because in any field, this is not going to change. No, not going to change. Same ANOVA table you will get, no matter whatever field of research you are working. Okay. There are four kinds of sum of squares. Type 1, type 2, type 3 and type 4. Type 3 are good for the unbalanced data. They are generated by using sequentially, developing the model sequentially. So I hope that is enough for this time. And we can discuss in detail, of course. I'll be happy to discuss this. And then type 1 sum of squares are good when your data is balanced. So we have done this analysis of variance. And you know this value, when this value comes below 0.05, this says that RIL effect is significant. What does that mean? That means the population what we have taken, in which there is some variation, like all people were not of same height or all people were not of same weight. There was some variation, like few were really shorter, few were really taller, something like that. Like you can imagine any phenotypic character and things would change here. So that the significant p-value means that. Well, now there comes several issues like permutation and the Bayesian inferences, so you can challenge this, that this is not a p-value, correct p-value. You may ask me to do permutation to get it the correct value, but let's settle down as of now that RIL effect is significant. This is the most common way to do the analysis. Okay? And so let's come to this below table. All of us understand R square of a regression? Yes or no? How many says no? Okay, one says no, two says no, three people no. Okay. R square is a measure of goodness of fit of your model, of your analysis. And this R square terms in regression, it may be efficiency, similar to efficiency in many other fields. Like when you drive a bike, you might know that the total fuel what you burn might be having 1000 calories. But the actual energy converted into the motion, may be only 30 to 40%. Now, well, 50 to 60, I don't know the modern engines. When I did my engineering, I know at that time, 10, 15 years before, 40% was a good enough engine. So, if you see that efficiency is 0.9, so what automatically suggests you? That it was a good fit? Though my breeding friend can always tell, R square 0.9 means fantastic. Okay, looks like very nice. If you see CV, how many of you know what is CV? Coefficient of variability. Can you explain this CV is good or not? Thank you, thanks. I need some few persons I need like this. They make the discussion lively. How many say this is what? Okay, well, Yes, yes. Well, see, I'm showing you a field experiment only. Okay, good. Thanks. I got three different views. So I got a view from there that CV may differ crop to crop. I'll say it may differ to experiment, even for the same crop. The way you are managing your experiment. Okay. Well, we'll come to that point. But this is a low CV, isn't it? Like it's a very good CV. Huh? And you see that R square is also equally good. So analysis looks good. And then if I see the mean value is 63 and the standard error of mean is only 3. So this clearly shows that analysis, data collection, everything is fine. So what is next step? Next step, those who know the breeding, is the QTL mapping. QTL means quantitative trait loci. That is a loci in the genome which is attributed to some of the quantitative phenotype. Like we were discussing for height. So then we could see that there are, let's not indulge into the method how we did it, okay? It's very simple actually. It's very simple. Just run a software. Huh? Well, not that simple also. So then you see that we got peaks at 4 places. So in the terms of QTL terminology, this we call LOD score, likelihood of odd, LOD scores. This is quite similar to the p-value. So where these peaks are crossing this 2.5, this line we also get through permutation. Okay. So whatever peaks are cutting this line are said to be a QTL. Right. So we could see that there are 4 places in genome which are responsible for height. So we were very happy. Students became very happy that yes, you could discover QTL, not only one, you could discover 4 different QTLs for the height. Now next step is to transfer. So but whenever we statisticians see any such data, we really do not get excited. Okay. On the other hand, we just open our eyes like this. Okay, let's stare into data. So now whenever data comes to me, we never put that data into analysis like this. Well, many of you think that we simply put the data in software, press the analysis button, result comes, we give it to you. So I asked my staff, why don't you show me the diagnostic plots? So then they come to me, they showed me a plot of data. It looks like normal, isn't it? So many of you will be really happy. Yes, data is normal. You know what is normal data? Anybody who don't know? Yes, the data which follow kind of normal curve. Yeah, like ideally any of the things in this world, in this universe, should follow a normal distribution because most of our assumptions are normal distribution. We can talk. Few people may say that there is not normality, but asymptotically we can always achieve normality. Any thought on this? Provocative thoughts? No? Because this is a point of discussion between the subject matter specialist and the statistician many times. Okay, so most of us agree that yeah, it has a very nice shape. So data looks good. Huh? But you know, a statistician never look for the distribution of the data. We look for the distribution of residuals. So when I ask them to, no, this is not acceptable, bring the distribution of residuals, this is what I got. Normality gone. We never look for residuals. Most of the time we only plot the histogram of the data. So then we suspected there is some problem. Then I advised them to run for the data diagnostics. Now this comes the data diagnostics, data curation. We ran another statistic, what we call Cooks-D, leverage, and there are several of them. We cannot really depend on any one of them. So normally we calculate three. One is called leverage, another is called Cooks-D, and three is the influence. These three measures we calculate. Based on that, I could see that on the extreme right, there is a peak. Well, we can see two peaks. So it looks there are two things which need some of our attention. If I would not have seen the data, if anybody could have normally analyzed the data, everything was fine. R square 0.9, CV is 4, R root MS square only 1% of the mean. Fantastic analysis, isn't it? Everything significant. 4 QTL you have got. What else you want? You can write even a PhD on that. But I can, yes, that kind of work. But we always come in between to a student and PhD. So then we said, no. Now there is some serious problem. Now it's my time to look into the data. So I looked into the data. I asked them to bring back raw sheet. And then while this is the open data, so this I opened the data sheet and then I looked for the observations. And then suddenly I observed that you could see that there was a peak after 400 observed number. I saw that this 442nd third observation for the real number 148, there were three blocks. In one block, yield was 71, another 70, it was 17.32. So now you could have guessed already what happened at that time. Data was recorded correctly, but when a student was entering into the computer from field book, since PhD we all know that we want to finish work very soon, he pressed 7 first, 1 first, then 7. This is a very obvious error, and we get such kind of errors in most of the data sets. But you cannot identify this error either by naked eyes, because we have here 440 observations and around 15 to 20 traits. The massive data we have and from several locations. So if you think by eyes one can capture, we cannot. We are also not magicians. But we run those diagnostic tools one by one, like we put on iteration and we forgot. Later on we see the influential observations and the plots, and then we try to conclude. So with no dilemma, we could convert this value into 71 points. Okay? It's clear? Okay. Again we did the analysis, like we repeated. Now program was ready. Before? You are talking for which, sir? Yes. This 39. Yes. This. Don't let's not. I'll discuss one by one. I'm talking for this first column only. When I corrected this column, then this is yield P2. Yield with two problems. This is yield P, yield with one problem. I'll show. Thank you for the question. So then we corrected it, and I corrected data entered into there. Yes, that is called statistical analysis system, SAS, and we do the most of analysis in SAS, and R also is a good system for that. Yes. So mainly we use both, combination of both. This plot we generated from them. It's a comprehensive process, of course. So then we change it into this, and now I analyze yield P as my dependent variable. So this is the case 2, analysis of yield P. No change, still they are significant. Okay. But if you see, R square has reached to 0.96 from 0.9. What you did? You just altered one observation. Because many of our friends say, oh, it's one observation. Let it take. Huh? That's not going to alter. But good improvement in your R square. That's not enough. Your CV is reduced by half now. Repeatability of trial is 2 times more than what it was earlier. Can you imagine that one observation can have that much of impact? Yeah, it can. If you see, overall mean has not changed. It was 63 point something, now it's 64 point something. So you really cannot see sometimes this into the summary statistics, what's happening in the analysis. It's very tricky. And if you see the standard error of mean, it is also reduced by half. It was 3 point something now. All of this magic is by only one observation. So I think now everything, yes sir.
So that was a typing error.
Mm-hmm.
So what if it was a typing error and you cannot go back and then recollect the information? Would you just guess and then put an average value? That would be cheating.
You are now directly going to the analysis. I will come to that also. The problem number 2 is same, what you are asking. So a very good question was asked. This time we saw the field book, and in field book we could see that actual value was actually 71. So we could do it. No problem. We sit with the supervisor of a student. We talked. Yes, this value was actually this. No problem. And I again did the QTL analysis. Okay. Now what I saw? All 4 QTL gone. All 4 QTLs gone. There is only one QTL. And this is also not the same QTL. If I put those plots side by side, this was for yield P, this is from yield P2. A new QTL has emerged. And all of these 4 QTL just gone. Like we have identified the wrong places in the genome. Implications? If you want to transfer this QTL in your further study, like for next of your student will validate them, they will disappear. You will never be able to validate them. And unfortunately, if you have taken a project to transfer these QTLs, you're gone. Problem? Only one observation. So that's why data curation becomes very, very important and key. When a data set comes to me for analysis, if I'm spending total 10 days, then believe me, out of 10, 7 days I put for data cleaning. We look each and every observation one by one, but different diagnostics. We simply cannot throw the data into software because statistical softwares are known to be GIGO, garbage in, garbage out. So the first figure is garbage, but second figure is okay. But you know, still I don't believe on my own analysis. I said, okay, let's see the plots. So we again saw the plot. It looks much better than earlier, but I will see the residual plots. Yes, now you got, madam. Oh, it is still same. Again, we repeated whole of the exercise what we had done earlier, and then I see that there is a gentleman lying in here. And there is a leverage also here, curve ratio we call. Oh. I again went into the data, opened the Excel sheet. Now I saw this time the problem was with the real number 107. Here if I could see that in block number 1 the value was 47.61. And please concentrate on only this. We are considering yield P, this I saw, and the observation number 19, 20 and 21, 300 and the same genotype. So there is a 50% reduction in the values. If I see that mean, so previously the mean value of that, because I have not changed of course, Well, I'll talk to this how mean changes. So now this is a trick. What can be the 47? This is certainly not a case of swap. We asked the student to bring the field books. He brought in, and what we saw in field book also it was written 47. Now problem becomes very, very complicated. It is a real problem what has been raised. What to do now? If we take all it is, the statistician is not agreeing. He is saying no, this particular observation is a problem. It is really taking all data to one direction. It is really changing the way you are looking to the data. So, what to do? Missing block technique is not a magic, sir. And we have done same in here. Like now we no longer, with the advent of generalized linear models, we don't use missing block technique because the way they adjust the technique, same has been taken care inside the general linear model. So it estimates the same value. So, no, if this kind of reduction would have been in all of the observation of block 1, there would not have been any problem. There is something very particular with this particular plot. There are several possibilities. There may be some toxic material in the plot. Or maybe, well, plants and everything, the plot has, because we don't have at that time photographs or anything left with us. We only had the values when we analyzed. Yes, but the related, so normally people harvest a similar number of plants, like 10 plants from each plot. So all those things were matching. So it took a long time to understand. Then there was another problem, what we thought that this is a bagging, seed labeling problem was also there. Because when we saw other related traits, all were drastically changed for this particular plot. So now it's clear that if we keep this data point in the analysis, it's not justice. So, but we cannot make it 87 also because that is also. So we simply remove this observation from further analysis. That's the why we take the replicates. Replicates are to ensure two cases. When you have loss of information, another to estimate the error. So we treated these observations as the missing value with a consensus. But it is a very tough decision, I can tell you. What of the Yeah, any observation, yeah. Yeah. Okay. What advantage do you see by doing that? Because you are not generating a new value. You are generating information you are not bringing from outside. You are extrapolating from the data what you have. So statistical software don't need that. They can automatically do that. Yes, so we did the same. We just put it as missing. Yes. Substituting that to an average is also misleading. Yeah. Yes. Even like if there are one or two, we can always treat them as missing. If there are many, then we have some other phenomena like transformations and other kind of things can be dealt with filters, applying some filters. But this case was clearly a case of misbagging, mislabeling. So we remove this particular row from the analysis. Okay? Is it clear? Now, we did analysis again. What we saw, now my R square has gone to 0.99. Okay? CV is 0.6. Error is 0.4. Yield mean unchanged. So can you see that how much confidence now you are gaining? Your standard error is 10 times reduced. Same mean. Like you are so much confident now on your own analysis. Just by one observation. Like now, of course, there are two. You have not done any extra effort. You have only taken some time, given time to a statistician to look into data and do the proper cleaning and analysis, rather than throwing your data into software. Sometimes breeder may not be impressed, or the subject matter specialist may not be impressed. Hey, you have only taken 1 or 2 values only. But those 1 or 2 values, we came to a conclusion after looking all of those 10,000 values. So that's why this takes time. But when you can conduct an experiment for 5 years, you should give at least 5 weeks or 1-2 months to its analysis also, especially to cleaning. Now, after all of this, this looks amazing, the results, the power, the statistical power, everything has increased. I again did the QTL analysis. We got a very, very high peak this time. This shows that how confident now we are for the QTL mapping. Log 6 around we are reaching now. It's a very, very high significance value for the QTL analysis. And out of 4 now you are getting only 1 QTL. Now I did the residual analysis. No change, but this time residual plot is also very nice. No problem. Okay? Things are good now. Now I was happy. I sent mail for the results are okay from my side. If you see the plots, leverage, curve ratio, Cook's D, also there's no problematic observation in here. So sometimes a single observation analysis can take weeks like this. But that's our aim, I hope. Whatever we are doing should be proper. So, Well, we cannot really say that. If you visit ICRISAT field sometime, we have got the precision fields. Well, I have worked for the ICR for 5 years. ICRISAT we have got the precision field, which means they are really precise. The fertility status, gradient, everything is very, very nice. The way we irrigate, those big tractors come and they irrigate from the top. Yes, yes. No, it's okay. I'm not saying you. But this kind of observations, if they are in your data, they'll make your 10% CV to 30% CV, or maybe 30% CV to 100%. We could see a 10 times reduction in CV. Sometimes most of the time if you see in the agronomic trial and even in the breeding trial, your CV reaches to 80%. That's true. I don't know if anybody says he has never seen a 80% CV. I have seen 8,000% CV. Don't worry. So we all are working with the real data sets. I fully understand your problem. It happens. But if you don't, so in these data sets, cleaning is still less important. In the data set where CV is very big, cleaning becomes even very, very important. Because then you are only playing with the error. You are not playing with the data, you are playing with the error. That's why sometimes your repeatability of experiments is very, very low. You do not observe same result next time or at the next location. Is it clear? So, if you see that how things change now. Now I will just show you some summary statistics from what I did. that I have taken this yield P2 with the first one, with two problems, yield P with the one problem, yield G is the good yield, like after removing those two problematic observations. So CV reduced to 0.6 from 0.8, error estimate reduced 10 times, R square also increased by 10% or more. This is a very important table, where our idea is to see which observation is different from which observation, like all possible pair comparison. If you see in the initial analysis, we have only 7,000 significant pairs. Out of total 11,175, so only 63% was the significant pair. All were other, around 37% were going to error, attributed to error. And minimum standard was 2.5, maximum error was 2.5. It was a balanced data at that time. Then, when we removed the one observation, 80% of our pairs became significant. Out of 63, like 17% more we have taken from the error by one observation. That 17% was controlling that yield variation from the trait. And the standard error becomes just 1 from the 2.5. When we remove both of the observations, this value becomes 10,000. And now 90% of our significant pairs, which were non-significant earlier, became. So you have taken out 20% from the error taken, and then model is attributing to that. What this costed you? A good analysis. Same data, repeatability is high, everything is fine. And if you see the QTL profile, these were the earlier 4 QTL, later on we gone 1, and when we did good analysis, we got even very high peak of the QTL. And one also small QTL just came in between. So this is one aspect of data cleaning, what I talked. Do you have any question? Those who survived. So we will not encourage any data without proper curation. And then of course, after once we curate the data, then the complexity of statistical analysis comes, what methodology we are selecting. Then that is the second level of complication that I cannot discuss as of now, what method we are doing. For that I run like 1 week long workshops on data analysis. So sometime we can join. No questions? Oh, yes. Thanks. I need at least one.
Do you make your data really available before cleaning it, or when you clean it before you can?
After cleaning. Data should be, because, okay. When, that's nice. So, yeah, when we share a data, we never share as a scientist. We share as an institution. Of course, credit goes to a scientist. There's a lots of credibility comes with the data. If you look, a data set is being shared by ICRISAT, or maybe the NIPGR, or maybe by your ICR institution, internationally, when it is on internet, anybody from the globe can access it. It has a name. You just cannot share a raw data which is uncleaned. Because my student who has recorded 71 as 17, and if I share it as it was raw and original data, no, that's wrong. So we need to share always curated data, clean data sets, because we are responsible for it. Tomorrow somebody is using the data for some analysis, and if he may not be very good in analysis, but at least the data set he should get correct. Best of our knowledge. Okay, you are touching a very sensitive issue. Now we may have a fight in here. So. First thing, I do not consider CV as a quality of trial. Again, two statisticians may not have same views. CV is a measure, but is not the absolute measure to judge the quality of experimentation, because few very good trials can have 8,000% CV. Few very good trials might have -1 as CV or -200 as CV. If you really want to see the repeat, many people relate CV with the repeatability of the trial. So then there are some measures for the repeatability of the experiment. There is a nice paper from FIFO in that. That repeatability of trials shouldn't be confused. Well, so CV is not the absolute measure. CV is just indicative that there is some problem. Okay? I discussed many of them. Well, I'll go with the significance of the model fit. Is the first one. Yeah. Model fit should be very good and significant. That's one way to judge. If you have low CV, but your model is not a good fit, repeatability will be low. And several other things like we check the repeatability. There are several measures to calculate repeatability itself in the literature. When we have this kind of dilemma, then we do that. We do not tell sometime to you, but we do ourselves, because whatever analysis is going from my desk, nobody should be able to raise questions to that. Lot score is something like p-value. So, but p-value should be low, lot should be high. Yes, yes. You know that already. So for the chi-square distribution, it directly corresponds to the p-value. Yes, sir.
As Dr. Rathore said, CD is sometimes maybe a misnomer. By and large it is, but not 100%.
Yes. Based on CV,
It
we
is an
shouldn't
indicator.
discard trial.
It is an indicator. But the best indicator is standard error of mu, or standard error. And I'm sorry, I came at the end of your discussion about CV, high CV. Was there a discussion about the high CV?
Mm-hmm.
Was there?
Mm-hmm.
Oh, yeah. High CV was, you were asking, no? Yes. High CV will have, you will observe high CV usually if the data do not follow normality. Usually you find that, Dr. Rathore, excuse me for my little knowledge about this, if the data follows either by nominal distribution or Poisson distribution, that may happen. For example, in pathology experiments or in entomological experiments or weed control experiments, you will find very high CV. And this CV has come from the data. So, for example, if you apply a herbicide, definitely the number of weeds will be very few. When there is no control, number of weeds will be high. Maybe, say for example, if you count the number of weeds in no weeding plot, it may be 300, 400. When you apply the herbicide, it may be 2, 3. So when you use that type of data, naturally the coefficient of variation will be high, because that data do not follow the normal distribution. That's why we have the transformation of data. I'm sorry,
No, like, I think, yeah, you...
Number two, in entomological data, big data, data, in field condition, data never be in a normal condition. Never. It can be only in glasshouse and laboratory condition only. If we release a known number of data, otherwise it is very difficult to have a normal data in field condition. And so we always allow even more than 100 CV, it is okay. If even after more than 100 CV, the treatments are effective, means it is effective.
Well, I can't say yes, I can't say no directly, because
it is the general resolution of the ICR that in entomology, plant pathology, weed control, if CV is high,
Yes.
you accept the recommendation.
no, I understand your point, but well, we cannot take an ICR guidelines as a general universal guide. I understood your point very well. In the cases like those kind of data, of course, we get a high CV. What do you need? Not mine, huh? Thank you. So normally, yes, I agree. We need not to relate the repeatability of experiment with the CV. But if CV is high, it is indicative that just look into your data. For a usual trait which follow into real domain from 0 to any real number should have a reasonable CV, should have. But many times the data, lack of data diagnostics, if you have come across some data which is really having that kind of CV, we can always discuss and let us see how CV do not come down. I analyze disease data in ICRISAT. We have lots of pathology experiments, but I understand those kind of data sets has really lots of variation, and we need to live with them. Like if you see I don't know how many of you work with the peanut in the aflatoxin, CV is usually 300%. You just cannot bring it down. It's normal. But the repeatability of the trials is high. But some characteristics, because we are not having an artificial world, we are living in a real world, as you mentioned. So in the field trials, sometimes we really cannot bring down CV below 100 also. That's very true. 100, sometimes 200, 300. I have several data sets. I know.
No, what I am in opinion, I am to say that if suppose treatment number X is found superior constantly for 3 years, even if CV is high, it's at par means at par. If it is found superior, means superior.
Yeah, if it survives in the pooled analysis also.
Pooled analysis also,
Yeah.
then it's okay.
Hmm.
And,
Yes.
yes,
And I.
And we should agree with the data of the scientist, because the CV and data in field condition is next to impossible. As earlier says, CV below 10 in field condition, especially in plant protection, it is always.
Well, giving a general statement is very difficult for me. I can give a statement based on data to data, and because of many of those things, and I really don't look for CVs. I look for the repeatability of trials. I look for other error. How much is the error in the data? Proportion of error to the total data variation. For me, they are more important than the CV. So you may like you might have seen those kind of data that I appreciate it, but I cannot give a general guideline on that it will be always 100%, it will be less than 100, it will be 500. So it really depends, and we need to really. But if you have done taken all kind of data cleaning measures, all kind of transformations you have tried,
Right, yes.
and then still it is coming, it's fine. Anybody will publish in this world, no problem for that. I 100% agree to that. Repeatability you can link to R square, otherwise there are some others, we can of course discuss them.